Okay. Sorry for the interruption. Let's continue. So we can use agents, poisons, toxins in medicine because many times the only difference between a poison and a medicine is just the concentration. So if you use it at the correct concentration, you can use it against a disease. In this case, for example, it is obtained from yew tree, the Pacific yew tree. So we know that in many cases the science and technology is inspired by the nature. It's a very useful tree. Of course, the high dose can kill you. Another form is this flower. Colchicine is obtained from the flower, and it binds to tubulin dimers and prevent polymerization. So here they stay as the monomer form, and these monomers cannot join the protofilaments and no more polymerization. So these microtubules, they will mostly depolymerize, so the cell is going to lose its shape, and somehow it helps this inflammatory disease. Most possibly the immune system cell cannot change shape properly. This is why colchicine is a very old and very valuable drug. It is also used in the FMF disease, the familial Mediterranean fever. Okay, and this is what your homework is about. You're supposed to find other examples of drugs, poisons, and toxins which target the cytoskeleton, and two of them, so that you're going to find, I think, nice examples. Okay. The nicest thing about the microtubule is they can be used for transportation. Why? Because their minus end is stuck, it is fixed on the centrosome, and the plus end growing this way, so it's all plus, and here. So because the direction is known, because the molecule walking on the microtubule can know which direction it is going, it can use the microtubules as a highway. And we know that they start from the center of the cell and they go till the end. Like there are bus lines starting from Mecidiyeköy and they go until the end of Istanbul. Just like that, they are useful lines, although they are very long. Okay, and they have two different movements. Either they can go outward, and we call them anterograde, and they can go backward, which is retrograde. Retro means back and away. For the anterograde, you can carry several things. The neurons are so long, diffusion is absolutely not an option. You can bring maybe mitochondria, maybe mRNAs to be translated here locally, some proteins, some neurotransmitters, these nerve proteins, any kind of protein and some material, they have to be transported from the soma, because we know that the genetic material, the inherited material, DNA, is here in the soma. So whatever protein is going to be produced, the production should start here in the nucleus. And then it can continue in ER, maybe in cytosol, and then can continue here. The translation can take place here maybe, or maybe here, or maybe here in the ER. But whatever, it should start from the DNA, and the distance between DNA and the nerve terminal is very large. This is why we need some proteins to carry them. Okay, there are some nice images of carrying the vesicles on the microtubules in this way, and maybe you all remember this figure. This is a motor protein. This motor protein is carrying a vesicle. Of course, the vesicle is much bigger than the motor protein itself. This is the cargo. It is carrying some proteins and some other molecules inside. And here you can see another motor protein carrying another cargo. And of course, it doesn't have to be neuron only. We know that any kind of transport between ER and Golgi, Golgi and lysosome, plasma membrane to lysosome, endosome, they are all happening this way. So all the vesicular transport that we have talked about, they don't happen randomly. They don't just diffuse and go back, or they just don't go anywhere in the cell, they have to follow these lines, and these lines are microtubule lines. And this kinesin protein knows the direction because there's alpha, beta, alpha, beta, alpha, beta subunits. So whenever it puts its steps on the surface, it can understand if it is going to the plus direction or minus direction. So if, for example, it started from the ER and if it is going to Golgi, yes, this is the right direction. And we have two different kind of proteins, motor proteins that walk on the microtubule, kinesin, which is going to the plus direction, and dynein, which is walking to the minus direction. So maybe this guy took something from the lysosome and it's taking it to Golgi, so it must be a dynein walking in the opposite direction. Okay. And especially kinesin has a very funny movement. It is consuming ATP. At each ATP it consumes, it makes a real step. So it walks step by step, like a funny guy. We can see that on the microtubule track. There are many different proteins walking, carrying stuff here and there. And the movement is mostly saltatory. The movement is saltatory because when it is moving on the microtubule track, at one point it is going to hit something and fall down. So let's check the microtubule structure. Here there are some attachments. It's going to hit this attachment, it's going to fall down, but when it falls down, it is going to find another microtubule and walk. This is very normal. This is why when you see a vesicle walking in the axon, it is going to walk in a saltatory way. It will walk and fall down, walk, fall down. Maybe it's going to find another. Okay. Somehow it comes until the end. Okay. So these are the kinesins. Oh yeah. Now, what keeps the organelles in their original place is the microtubule cytoskeleton. Now, this cytoskeleton fixes the organelles in their place. It helps their transportation. But it's not only them, it's also some motor proteins pulling them. We know that in the cell, ER is close to the nucleus, Golgi is a little bit far. If you get rid of all the motor proteins and all the microtubules, what's going to happen to Golgi is that it is going to splash here and ER is going to collapse on the nucleus. So of course, again, this is reversible as the microtubule is taken back. If the microtubule is recovered, they are also going to take them back to their original positions. So the Golgi will come back to the middle of the cell, somewhere middle, and the ER will go back to its original position, a little bit far from the nucleus. And this is with the help of kinesin and dynein motor proteins. Okay. These motor proteins and microtubule is also very useful for movement. That was the starting point of our chapter. We use our muscles for movement as the body, but within the body there are also other movements. The liquid, for example, from the respiratory tract, in the brain, in the intestines, they're moving. They have to move. And for them to move, they are pushed by cilia. What happens to cilia is it's in this original position, it comes back and shlak, comes back, shlak. Okay? So it waves between these two directions. And in this, to be able to move in these two directions, for a cilia, there is a microtubule organizing center at the bottom of each of them. And with the help of the motor proteins and the little bit of microtubules here, they can wave and they can push the liquid, they can push the fertilized egg to the uterus, for example. The other element of movement is flagellum. we don't have much cells with flagellum, just the sperms. In the unicellular cell environment, they have many more flagella containing cells. Movement is the key for them. This is how they hunt. This is how they find their food. For us, this is how we reproduce. The sperm must be healthy and mobile, otherwise it will not be able to find the egg and fertilize it. Now the microtubule structure in flagellum is a little bit different from the microtubule structure in the rest of the cell. It doesn't have these 13 protofilament anymore. Well, it has it, plus it has this half croissant shape protofilaments. And you have 1, 2, 3, 4, 5, 6, 7, 8, 9 of them, plus 2 of them in the middle. This is called 9+2 organization. In each case, there are motor proteins on the surface. And these motor proteins allows the microtubules to move themselves against each other. For example, in this case, there are ciliary dyneins. And these dyneins, when the energy is given, they walk on the surface of the other microtubule dimer, and this is not a real dimer. Remember, it is the structure. Okay, it is walking on this, so it will push the shape, and when it is released, it will come back to the original position. Okay. It can slide this way, or it can bend this way. It depends on if you have these linking proteins or not. Okay. And we know that there are different kind of movements in this way. The sperms move by waving their tails, and the cilia is rather like bending. Okay. And there was a seminar in our department just before the pandemic. It was about the nanorobots, and the strategy used in nanorobots is exactly this. They are mimicking the flagellum to mobilize their robots. Okay.